Okay, morning everybody. So let's get started. I think we finished the digestion of carbohydrate and protein, right? Last class, and this was last class we talked about. I mean, you can go over again, just summarize the protein digestion absorption that we talk about, the enzymes, and and the location that the proteins are digested and absorbed. Remember, so amylase and the other glucose. Sorry, I kind of messed up with the carbohydrate metabolism. The enzymes, right? I mean, digested, and then eventually protein will be break down. The polypeptide will be break down to oligopeptides and di and tripeptides, and the anthracytes also secreted endopeptides and carboxypeptides, right? And to further break down the dipeptides and the tripeptides, and these peptides eventually break down to free amino acids and be transported into the portal vein. And also transport of amino acids requires the transport system, right? And we just mentioned the sodium dependent and sodium independent transporters. You don't need to memorize the details, but just know that protein transport also requires transporters. Now let's talk about lipid digestion and absorption. So what are the most abundant lipids present in our food, in the diet? Do you guys know what are the most abundant lipids in the diet? Triglyceride, right? Triglyceride is the most abundant lipids in the diet. And also we have a diet, food have a cholesterol and cholesterol ester and some phospholipids. But again, predominant amount of the lipids in the diet is triglyceride. So here is the triglyceride structure. I think it's important. Try to remember this structure will help you understand later, how lipids metabolize or synthesize. So basically, triglyceride has a glycerol backbone, right? And 3 fatty acids attached to the glycerol backbone to form triglyceride, right? And glycerol backbone has 3 different carbon at a different position. We call the first one, the top one is SN1 position carbon, and middle one is SN2, and bottom one is SN3 carbon, right, in the glycerol. So, and also 3 different fatty acids attached to the glycerol backbone are different in terms of their saturation. For example, the SN1 position and fatty acids at the SN1 position normally is saturated fatty acids, right? And SN2 fatty acids usually is monounsaturated fatty acids, and SN3 can be polyunsaturated fatty acids, right? So different fatty acids attached to different position at the carbon in the glycerol. And this just gives you a structure of other lipids, for example, cholesterol, like and cholesterol esters. So the difference between cholesterol and cholesterol esters is cholesterol ester has a 1 fatty acid molecule attached to the cholesterol molecule, right? So cholesterol ester again has an additional fatty acid molecules attached to it. And phospholipids, the type choline molecules, you can see also basically it's similar to the triglyceride, right? This is again the glycerol backbone, 3 carbons, and SN1, SN2, 2 fatty acids attached to SN1 and SN2, and SN3 position there is the phosphor group, right? And choline molecules attached to phosphor group, and then the SN3. So the only difference is the SN3 position part, right? And also some concepts about free versus esterified lipids. They also are different molecules, but they are very similar, right? So lipids, for example, triglyceride is considered esterified lipids because 3 fatty acids attached to glycerol backbone, right? And 3 free fatty acid molecules are considered free lipids, right? So, and similar to the cholesterol esters is considered esterified lipids, and free cholesterol is a free lipids, right? So something, the lipid molecules, if they attach to, the fatty acids, and then it's considered the esterified lipids, right? Is that clear? So again, free fatty acids is free lipids, and cholesterol is free lipids, and cholesterol ester with the free fatty acid attached to the cholesterol is esterified lipids, and also triglyceride, 3 free fatty acids attached to glycerol backbone is also considered the triglyceride, the esterified lipids. Okay, so lipid digestion. First question we always wanted to ask is that where does lipid digestion occur? It's begin in the stomach, and most importantly, small intestine is the main site for lipid digestion, right? And the 2 enzymes that are very important to break down triglycerides, the lipase. So gastric lipase and pancreatic lipase. So by the name you can see that gastric lipase is secreted in the stomach, right? And pancreatic lipase is secreted by pancreas. So these two are lipase and does the same thing to break down triglyceride. And gastric lipase, these two enzymes, although they break down the triglyceride, mean that they remove fatty acids molecules away from the glycerol backbone. The two enzymes have a different preferences because gastric lipase normally cleaves the SN3 position, right, of free fatty acids. So gastric lipase usually removes fatty acids from SN3 position and generating 1,2-diacylglycerol and free fatty acids, one molecule free fatty acid. So this one molecule free fatty acid usually comes from SN3 position, right, in the triglycerol molecules. And pancreatic lipase cleaves SN1 and SN3 position, right, and generating 2 monoacylglycerol and two free fatty acids. Right? Does that make sense? So if glycerol molecules, one glycerol molecule is hydrolyzed by gastric lipase and only generate one free fatty acids, and this free fatty acid is cleaved from SN3 position, right, in the glycerol molecules. And if one glycerol molecule is digested by pancreatic lipase, usually releasing the two free fatty acids. These two free fatty acids from SN1 and SN3 position. Yes?
What does it mean,
TAG, oh, triglyceride. TAG, triglyceride. Just initial issue. All right. Any question? And we also have a different enzyme that digests phospholipids and cholesterol, right? So phospholipid lipase A, which is secreted by pancreas, and phospholipase B, normally present in the brush bottle. These two phospholipase, usually hydrolyze phospholipids and release the release fatty acids and lysophosphatidylcholine. And cholesterol esters, it's esterified lipids, right? That has one fatty acid attached to the cholesterol, and cholesterol esters can be digested or break down by cholesterol esterase, which is also secreted from pancreas. So add cholesterol esterase and hydrolyze cholesterol esters and release free cholesterol and one free molecules of free fatty acid. All right. And absorption. So if you still remember when we talked about last class, this is the enterocyte. Remember that on the top of the brush bottle membrane there is called unsterile water layer, right? Present. So because of this unsterile water layer, that makes the lipids kind of prevent the lipids penetrate through this water layer and into the enterocytes. So if free lipids without any facilitation of the vesicles, these free lipids were not able to efficiently absorb it into the enterocytes, right? So, but our body was able to secrete bile acid, remember? So bile acid is a very important, together with the lipids, form so-called micelles or vesicles, right? And this structure is very important, can significantly increase the efficient absorption of lipids into the enterocyte. So that means, so the lipids molecule usually have a hydrophobic and hydrophilic portion, right? So with this bile acid help, so the hydrophobic portion of the lipids is buried inside of these vesicles and expose the hydrophilic portion outside the vesicle. So hydrophilic portion, so make these vesicles hydrophilic, so then can penetrate the water layer and enter the enterocytes, right? So this is very important. The bile acid actually play a very important role to facilitate lipid absorption. So sometimes, some kind of disease, patient have a problem with the secretion of a bile acid, then also have a problem with the lipid absorption. Okay, and transporter of the lipids. Once it's absorbed into the enterocytes, and an interesting thing is it's different from the carbohydrate, glucose, and amino acids. The lipids eventually, first of all, they not directly enter the circulating bloodstream. They actually, first of all, they transported to the lymphatic system, right, and eventually to the circulation. So this is something different between lipids and the protein and carbohydrate absorption. Another important thing is I wanted to mention that the lipids, particularly fatty acids, right, eventually break down fatty acids from the triglyceride. So fatty acid, once is absorbed into the enterocyte, and they immediately re-acidify back to the triglyceride. So that will make the free fatty acids level inside of enterocytes lower, right? So this process will keep free fatty acids lower in the enterocytes. So it makes concentration gradient. So that means outside of the enterocyte have a higher levels or high concentration of free fatty acids and lower concentration free fatty acids inside of enterocytes. So it makes a concentration gradient. This concentration gradient will help more lipids absorbed into the enterocytes, right? So again, once the fatty acids absorb into enterocyte, immediately re-acidify it back to the triglyceride and keep free fatty acid level low inside of the enterocytes. And other thing, the second thing is important, that the fatty acids, once it enters the enterocyte, usually is transported by so-called fatty acid binding proteins, right? So fatty acid binding proteins, they bind fatty acids and help fatty acids transport fatty acids to the places where fatty acids re-acidification occurs. For example, ER, endoplasmic reticulums, right? ER. ER is a place where the triglycerides is synthesized, right? All right. So yeah, I think I talked about the export a little bit earlier than something happening in the enterocyte. So again, fatty acids, first of all, is transported to the the lymphatic system, right, and then the bloodstream. And also important thing is the transport of the fatty acids into the circulation requires is called a lipoprotein system. We're going to talk about it later. We have a specific session about lipoprotein. So lipoprotein is very complex structure because fatty acids, free lipids is not water soluble, right? So you cannot, freely transport it in the circulation. So you need something to help lipids transported from between tissues. So lipoprotein is the system. That main function of lipoprotein is to help transport lipids between tissues. So in the enterocytes, once the lipids is synthesized, is absorbed and reacidified to triglyceride and packed into the chylomicron. So chylomicron is one of the lipoproteins that primarily made in the enterocytes, right? In the small intestine. So chylomicron can help transport the triglyceride, which initially absorbed it from the food, and to the adipose tissue for storage, and also can transport it to the liver for further metabolism. Okay, so this is just a diagram show you, a little bit of detail that I just talk about it. Again, fatty acids and break down from the dietary triglyceride, right, and absorb it into the enterocyte, and immediately be reacidified back to triglyceride, right? So keep free fatty acids concentration low and to allow the more fatty acids being absorbed into enterocyte. And once triglyceride is synthesized, reacidified, then packed into the chylomicron and transported into the circulation and to the other tissues. Any questions about lipid digestion? So, so now we finished think about digestion, absorption of 3 macronutrients, right? And there are actually some common things and also different things. So let's talk about the differences. So carbohydrate and lipids digestion usually occur in the lumen or small intestine, and protein digestion is also in the stomach, small intestine, but the difference is that the enterocytes also play an important role in terms of digestion of protein. I remember enterocytes secrete 2 important enzymes, aminopeptidase and carboxypeptidase, so can further cleave the break down the di and tripeptides, right, inside of enterocytes. So this is something different, and usually for carbohydrate and lipids, we already finished the digestion in the small intestine and absorbed it into enterocytes. And lipids digestion, lipids absorption is kind of also special because it's of the hydrophobic nature of the molecules, right? So it requires bile acid to facilitate lipids absorption into enterocytes. And also the differences between lipids and other 2 micronutrient absorption, transport absorption, is that the lipids usually will transport first to lymphatic system and then the circulation. And also the fatty acids once it enters the enterocytes and reacidified back to triglyceride, and requires a lipoprotein, right, to help transport it to other tissues. Yeah.
What was the part of the lymphatic system with the lipid?
The lymphatic system.
Yeah, something that you mentioned.
Right.
Yeah, what was its already infection?
Well, I mean, lymphatic system is, also kind of like similar, like, well, not similar like the bloodstream. I mean, lymphatic system, you transported lipids into the lymphatic system, and then through the lymphatic system eventually enter the circulation, right? Okay, any questions about lipid digestive absorption? No? All right, so now let's talk about carbohydrate metabolism. So carbohydrate metabolism 1 and 2. So in this session, we're going to talk about the general concept of glucose metabolism and touch base the glycolysis and maybe TCA cycles, right? So general concept of glucose metabolism. We're going to cover what type of metabolic pathways that involve glucose metabolism, and also talk about the glucose homeostasis, means that our body has all the regulatory system try to maintain our blood glucose level at the certain range. And we also want to talk about the glucose transporter because this is a very important process that can maintain the blood glucose level and glucose metabolism. And we wanted to talk about the glucose metabolism in specific tissues because different tissues have different function or they metabolize glucose differently, right? For example, liver versus the adipose tissue, right? So liver is the very important tissue that can synthesize glucose, right? Can also break down glucose. It can also do the gluconeogenesis. And adipose tissue usually, not able to do glycogen synthesis, gluconeogenesis. So different tissues metabolize glucose differently. This is one concept. And the other concept is that the even in the same tissues, and metabolic activity can be different in a different physiological condition, right? For example, liver, under the fat condition, liver usually just does the glycogen synthesis, right, to try to store carbohydrate in the form of glycogen. So glycogen synthesis activity will be very active, very high in the fat condition. But gluconeogenesis activity will be low because we don't need to make glucose, right? We have enough glucose from the diet. So, but under the fasting condition, we don't want to synthesize glucose, right? Synthesize glycogen. So we want to break down the glycogen for energy use, and we also want to make a glucose, so the gluconeogenesis, to maintain blood glucose level. So again, try to keep in mind that metabolism can be different in different tissue and can also be different in the different physiological condition. Here we mostly focusing on the fat and fasting condition, right? Okay, so digestion, absorption, and transport carbohydrate, we already covered this, right? I just wanted to remind you again. So final product of digestion, you guys already know, right? What are the three sugar molecules that are eventually can be absorbed? Glucose, fructose, and galactose, right? Okay, so three final product of a carbohydrate digestion that can be absorbed into the body and metabolized in the body. And I just want to mention the glucose is the most abundant of a carbohydrate, right? Absorbed. They have much more high levels compared to the fructose and galactose. And second important thing I want to mention that the cellular fuels, our body needs energy, right? The cells need energy for activities, and energy fuels. I think we mentioned earlier, right? In terms of the nutrient level, we know that carbohydrate, protein, and lipids, right, can be utilized to make ATPs as energy. But if we come out, a little bit more details and a little more like lower levels, the molecules that can be eventually absorbed into the body and into the cells and metabolized in the cells and produce ATPs are glucose, fatty acids, ketone bodies, amino acids, and other gluconeogenic precursors like glycerol, lactate, propionate, right? So only glucose, fatty acids, and amino acids we know actually it comes from the diet, right? We digest food and absorb it into the body. But ketone bodies and glycerol and lactate and these kind of molecules actually is metabolized, right, from these nutrients, and body make these molecules from the nutrients that we absorb, right? But these are all important energy fuels. That means these molecules can be oxidized in the cells and produce ATPs. And glucose is primary metabolic fuels in the humans in terms of energy production, right? So for example, in our diet, 32 to 37 of energy that we need actually comes from the glucose, right? So glucose is very important energy fuels in the humans. And all tissues are able to use glucose as energy fuels. So glucose have several different metabolic fate in the different tissues. I already mentioned a little bit, right? Different tissue can metabolize glucose differently and depend on, again, the physiological conditions. And physiological state, again, determine, the metabolic fate, right? Fat fasting can make a glucose metabolize differently in different tissues. I already give you an example, right, in the liver. So these are the very important concepts that you need to keep in mind when doing, learning, when you learn the metabolism of nutrients. So how your body metabolizes carbohydrate, and we also need to understand this metabolic process at the tissue organ level, and also at the subcellular level. So at the tissue organ level, I already mentioned, right? Because different tissue again metabolize glucose differently, and at the cellular levels, because some organelles are very important for metabolism. For example, mitochondria. You must be very familiar with mitochondria, right? So mitochondria is like a power plant, right? It's an important place that oxidize nutrients and for ATP productions. So and some other organelles like ER mentioned is a place that actually important for synthesis of a triglyceride. So different organelles does a different function in terms of metabolism, right? Okay, more important general concept about the anabolic pathways, catabolic pathways, and metabolic pathways. So anabolic pathways usually is these pathways that synthesize larger molecules from the smaller molecules. Okay? For example, glycogen synthesis. So glycogen can be synthesized from the small glucose molecules, right? So glycogen is very complex, molecules. So this process we usually think is the anabolic pathway. And catabolic pathway is pathway that break down larger molecules into the smaller molecules, like glycogen breakdown pathway, right? Or the triglyceride lipolysis pathway. You break down triglyceride molecule and release fatty acids. And every anabolic pathway, this pathway can link between an anabolic and catabolic pathway. For example, TCA cycle. So TCA cycles is a very important process that connect the anabolic and catabolic metabolism. And also another concept about the anaerobic pathway and anaerobic pathway. So aerobic pathway, that means that pathway that requires oxygen or air for the process. For example, the oxidation process, right? Glucose, fatty acids, and amino acid can be oxidized, and this oxidation process requires oxygen. And this oxidation process normally occurs in the mitochondria, right? Mitochondria is very important organelle that does the oxidative metabolism. So, so this process considered as the aerobic metabolism occurs in the mitochondria and requires oxygen. And end products of aerobic metabolism should be, what do you guys think? The end product of aerobic metabolism, some nutrient can be completely oxidized to just similar, the chemical reactions, oxidation reaction. It'll be CO2 water, right? Okay. So anaerobic pathways. Anaerobic pathway, that means this metabolic pathway does not require oxygen, even can occur without oxygen. Okay, and it also means that this anaerobic metabolism also does not need mitochondria. Right. So it usually occurs in the cytosol. An example of anaerobic metabolism pathway is glycolysis. Right? Glycolysis of glucose metabolism. It occurs in the cytosol and does not require oxygen, does not require mitochondria. An end product of the glycolysis, for example, anaerobic metabolism is, does anybody knows? Pyruvate, very close, yes, pyruvate. Pyruvate is a product of a glycolysis. Under the anaerobic conditions, pyruvate will be eventually, yes, converted to lactate. So lactate is the final product of anaerobic metabolism, glycolysis, right? Okay. All right. Okay, let's just finish this table. We can take about 10 minutes break. So what do you guys need to know for this metabolic metabolism, glucose metabolism is, I have actually provided two slides of tables, right? First of all, you need to know the pathways that we're going to learn for glucose metabolism. We have 5 different pathways we need to learn, like glycolysis, glycogenesis, or also called glycogen synthesis, right? Glycogenesis or glycogen synthesis, and gluconeogenesis and glycogenolysis. Glycogenolysis also called glycogen breakdown, right? Glycogenolysis is also called glycogen breakdown. And finally, the pentose phosphate pathway. So we're going to learn 5 different metabolic pathways of glucose. And what you need to remember is, first of all, the rate-limiting reactions. So you don't need to remember every step of the metabolism, metabolic pathway, every step of the reactions. You just need to remember the key rate-limiting reactions, right? And key enzymes that characterize this rate-limiting reactions, right? And how this key enzymes is regulated by hormones, for example, insulin, or glucagon, or whatever other important hormones. Oops! Oh, I'm sorry. I talk a little bit backwards. So you also need to know the pathway, whether they are catabolic or anabolics, and whether they are active in the fat condition or fasting condition, right? And in which tissues this pathways occurs, and what is the main function of this pathway? Why our body needs to do this metabolic activity or pathway, right? For example, I just take let's say the gluconeogenesis, right? So gluconeogenesis, do you guys know which tissues are the main tissue that can do the gluconeogenesis? Liver, right? And what is the other tissue that can also do the gluconeogenesis? No. Kidney, right? So liver and kidney, you can do the gluconeogenesis, and not the other tissues, right? And what is the function of the gluconeogenesis? Why we need to do gluconeogenesis? Why? Or I can ask a different question. So in which physiological conditions gluconeogenesis is active? Fasting, right? Okay, so why we need to do the gluconeogenesis during the fasting? What is the purpose of a gluconeogenesis? To make a glucose, right? So the purpose of gluconeogenesis to make glucose and to raise blood glucose level, to maintain the blood glucose level, is the main function and purpose of gluconeogenesis. So something like this, right? So you try to remember during the learning process of the metabolism. Okay, so now let's take about a 10 minutes break and we'll come back 11:35.
Right, we should know this from biology in a way.
Why do you need all that potassium slash codeine bullshit? I'm working out though in a lot. Like you're working out, right? Right. This. One hand is this. Well, I guess you're ready to your part, so. Oh, you mean variety. You mean variety. Yeah, you mean variety. I would just laugh. Crazy. Yep, you switch every week, but you go I don't know. I said at 7 a.m. I know. Sat down. I mean, like, as someone was there, I didn't. Why didn't you Oh, really? Oh, I'm doing that. Are you kidding me? And you telling me to do it? Well, yeah, I'm do it. Did we get yelled at? Yeah. I mean, a lot of my well, she was staring at Oh, we literally both threw up the deuces. Throwing up the deuces. Oh, I like that one. I use that antibiotic to really see Hi. Good job. How you doing, Julia? Hey, guess what? I was driving on, what's it called? When you get off of the 280, like Washington. And you were right there. Where you live? You were like waiting on the sidewalk early in the morning, like at 8. Yeah, 8 a.m. You're waiting for the Because I was pointing to the yellow light. There you are just sitting there. And I was like, see ya. I was late though. I was so late to my 8:05 class. I thought 7:20. I forget I'm not a morning person. I forgot that. I was like, it was like 8:20 when I was at my first class. It's Glenn's lab. Right. There's some 8:05 to 10:20. He was on at 8:45. So I had to go get coffee. I was like, I didn't have it. The whole time. As I was writing up, I didn't even look over to your direction. What? Like when you're taking access to the code from the body, it doesn't
Okay. All right. So let's move on. Circulating glucose. Oh, sorry. All right. Can anybody hear me? Okay. So circulating glucose levels, very important, right? We always want to maintain the glucose levels at very narrow range. I don't want to let it go high and go low. So usually when we eat, we have a higher glucose level, right? After eating, and then when we're fasting, we have a lower glucose level, but always wanted to maintain between this range, right? 5 and 3.9 or something like this. And but if you ingest a high carbohydrate diet, definitely will have a little bit higher blood glucose level. But our body can adjust to lower glucose to maintain it in very narrow range. And the reason is important that if we blood glucose is too low, and definitely is not healthy, right? We know that we have some problems. So because of the brain is heavily relying on glucose for energy metabolism. So mostly because of the brain. So we wanted to maintain the normal levels of glucose. But too low, and that's not good for brain. Okay, so now let's talk about glycolysis. It's the first Our important pathways of glucose metabolism, glycolysis. So the glycolysis is a metabolic pathway that can convert glucose into pyruvate. Again, also depend on the, whether it's aerobic or anaerobic condition, right? And the aerobic condition, glycolysis pathway will convert the glucose to pyruvate. And gluconeogenesis, I think we just wanted to repeat it, this different pathway again. Yes. And so glycogen genesis or glycogen synthesis is a process of glycogen synthesis, right? And gluconeogenesis is a metabolic pathway that can make glucose from non-carbohydrate carbon substrates. And glycogenolysis or glycogen breakdown, basically the process that breaks down glycogen to release free glucose into the circulation if it happens in the liver. But if it happens in the muscle, glycogen breakdown usually does not release glucose into circulation, but instead utilize glucose for energy within the muscle. Right? We're going to explain the reason why muscle not able to release glucose and liver is able to release glucose from the glycogen breakdown process. Right? Now let's talk about the glucose homeostasis. Homeostasis means that we always want to maintain blood glucose level at the normal range either under fat or fasting condition, right? So on fat condition, we don't want our blood glucose level goes too high after eating, right? So you have to metabolize glucose and to maintain glucose level. Or the fasting condition, we don't want our blood glucose to go too low, so we break down some stored carbohydrate, and we also make glucose, right, to raise the blood glucose level during the fasting. So let's talk about in the fat condition how our body is able to maintain blood glucose level and not allow the glucose level to be too high after eating, after meal. So in the fat condition, when we eat food and the carbohydrate in the diet, we know that can be digested in the small intestine, right, absorbed it into the body. And some of the glucose can metabolize just in the small intestine because intestine also need energy, right? So some of glucose can serve the energy for small intestine and metabolize in the small intestine. And the majority of the glucose being absorbed and into the body. And 20% of absorbed glucose goes to liver for metabolism, right? And the rest of the glucose, either non-uptaken or non-metabolized glucose, actually goes into the circulation and provide to the other tissues in the body, right? So let's look at how glucose is metabolized in the liver. We know in the fat condition, the most important process metabolic pathway can be activated in the liver is what are we going to do with the extra glucose we absorb it from the diet? Yeah, stored in the energy in the form of glycogen. So the glycogen synthesis pathway or glycogenesis pathway will be activated in the fat condition, right, to store the glucose that absorbed from the diet in the liver. And also liver need to use glucose for energy, right? So glycolysis pathway is also activated. So glycolysis basically, oxidized glucose either can be complete oxidized depend on the tissues, but in the liver it can be complete oxidized. So most of the glucose absorbed into the liver can be synthesized to glycogen stored in the liver, or can be oxidized for ATP production in the liver. But liver can also do other two metabolic activities, right? For example, gluconeogenesis, glycogenolysis, or glycogen breakdown. And these two pathways, what do you expect would be the activity to the activity in the fat condition? Glycogen breakdown, gluconeogenesis. Do we need the extra glucose in the fat condition? No. So these two pathways should be lower, right? Activity should be lower. So we don't have much, glucose being made from gluconeogenesis, and don't need too much glucose be breakdown from the glycogen. So in the fat condition, glucose is metabolized through glycolysis and the glycogen synthesis. Right? Does that make sense? Right? And then in terms of the glucose that enters the circulation, it can go to the different tissues for metabolize, right? So you see 22%, a lot of glucose in circulating glucose actually uptaken by the brain and used as energy in the brain in that fat condition. And 2 to 3% go to other tissues. And the important thing that is the other two tissues, muscle and adipose tissue, they can also take a lot of glucose. And I also wanted to mention that, remember that the glucose transporter we talked about early, right? When we talk about digestion absorption. So glucose always being transported into the cells by transporters, and different tissue cells, they have a different type of glucose transporters. So for example, in the brain cells usually have GLU3 and GLU1. The GLU3 and GLU1 are the most predominant transporters in the brain. So these two transporters can transport glucose from circulation into the brain cells for energy production. And in muscle and adipose tissue, these two tissues are so-called insulin responsive tissue. And because these two tissues, the predominant transporters in these two tissues is GLU4. GLU4 is an insulin-dependent glucose, meaning that GLU4 activity requires insulin stimulation. So insulin can stimulate GLU4 activity on the cell surface and then promotes glucose transporter into these two tissues. Right? So in the fat condition, the other thing is important is the hormones. So which hormones do you think the level should be increased in the fat condition when we eat the food? It's already in the slide. Insulin, right? Insulin is released from the pancreas, and insulin secretion is increased in response to the glucose. Right? So glucose is the triggers of insulin secretion. So when our blood glucose level increase, and that can trigger insulin secretion from pancreas. So insulin level is also increased because of increased blood glucose level. And insulin can stimulate glucose uptake by GLU4 into muscle and adipose tissue. Right? So because of lots of glucose either being retained in the liver as a glycogen or uptake by these tissues, so blood glucose level will be decreased, lower, right? So again, this activity, particularly muscle and adipose tissue glucose transport activity, is very important. So in the diabetic patients, because of they have some problem with the insulin sensitivity, so these two tissue will not respond to the insulin, so they will not effectively transport glucose from circulation into this tissue. So as a consequence, the blood glucose level remain high, particularly when after meal, right? So diabetic patient with high glucose level because of dysfunction of this process. So this is how our body try to maintain blood glucose levels, right? So you see all these things, work together. All these tissues work together to reduce the blood glucose level after meal to maintain the normal range, right? And how about during the fasting? And because we will not have glucose from the food, right? We don't eat food. So how our body will do something to not allow the glucose level to drop too significantly. So several tissues and metabolic pathways that involve in the regulation of the glucose levels during the fasting. First, liver, of course, is important, right? During fasting, what liver will do, you guys can guess, which pathway will be increased in the liver during the fasting. We have a lower glucose, right? We need the glucose. Glycogen breakdown, right? And gluconeogenesis. So during the fasting, liver can break down stored glycogen and release glucose into circulation, right? And try to raise glucose, blood glucose, and also liver can be gluconeogenic, can make a glucose and release glucose into circulation. And also I mentioned the kidneys is also the tissues, the organs that also can make a glucose through the gluconeogenesis, and this only under the prolonged fasting, right? So these two tissues that involved in the regulation of a glucose metabolism basically try to contribute glucose to the circulation, raise the blood glucose levels, right? And why we don't have a muscle in here, because muscle also have a lot of glycogen store, right? I already mentioned that muscle be able to break down glycogen, but only use for itself, not release into the circulation direct. Because of muscle lacks of the key enzyme, glucose 6-phosphatase. We're going to talk about later, right? Glucose 6-phosphatase is the key enzyme for glycogen breakdown for making free glucose, right? And also the very key enzyme in the gluconeogenesis. Okay, so this is how circulating blood glucose level can be maintained during the fasting condition. Again, liver and kidney are the two tissues, organs that involve this regulatory process by break down glycogen or making glucose through gluconeogenesis, right? Okay, now let's talk about glucose uptake by tissues. We know that glucose uptake is very important, right? I mentioned if something's wrong with the glucose uptake, then you have lots of serious metabolic problems. Like diabetes, right? Diabetes, patient usually have a lower group 4 levels in the muscle and lower glucose transport activities in the adipose tissue. So is a key regulative step controlling glucose homeostasis. So we have, so far we have actually a lot of different glucose transport have been identified, but here we just wanted to focusing on the 5, right? Group 1, 2, 3, 4, and 5, and different tissues that express or produce a different glucose transporters, right? And in general, we just wanted to divided this glucose transport into 2 categories. One is called insulin-independent glucose uptake, like group 1, 2, 3, and 5. And these 4 different type of glucose transporters transport glucose independent of insulin. So insulin does nothing to regulate this transport activity by this 4 type of transporters, right? And the second category, which is very important, is called insulin-dependent glucose transporter, group 4. I mentioned, right? Group 4 is the transporters that the activity is only when insulin level is high. So insulin can stimulate group 4 activities and stimulate glucose transporter by group 4. So 2 categories you try to remember, right? And this table just gives you some idea, the different type of glucose transporters and the major site of the location of this different type of transporters. For example, group 2 usually is present in liver, pancreas, beta cell, and kidney, small intestine. Right? So liver, pancreas, beta cell is a major site that express the group 4 and group 2. And group 4 usually skeletal muscle, cardiac muscle, and cardiac muscle, and adipose tissue are the major tissue that express the group 4. And group 5, do you guys already know, right? We have learned last class. Small intestine, remember. So in the enterocyte, right? Fresh border membrane. Group 5 usually transport fructose, right? So group 5 is a fructose transporter. And SGLT1, we also learn about this transporter, right? It's also in the small intestine and also kidney, but it's responsible for glucose transporter from small intestine lumen into the enterocyte. So just try to focusing on these 4 different type of transporters. So how glucose transporter regulate glucose metabolism? In fat condition or in fasting condition, we also always talk about these 2 different physiological conditions, right? So in fat condition, 2 main glucose transporters are involved in the glucose metabolism, group 2 and group 4. So group 2, we know which tissues, organs that express mostly group 2. We just talked in last slide. Do you guys remember? Which tissues? You can look at your notes. Liver and pancreas, beta cell, right? So group 2, okay, and group 4. Group 4, which tissue? Skeletal muscle and adipose tissue, right? Thank you. Yes. So and insulin dependent glucose transporters through group 4, right? Group 4. And here just give you kind of numbers, so you kind of sense how important the group 4 in the glucose transporter in muscle and adipose tissue. So 80% of blood glucose is transported into the skeletal muscle under the fat condition because of a high activity of group 4 that is insulin dependent, and then 20% of blood glucose level that respond to insulin that transported into adipose tissue. So muscle is very important tissue that takes up lots of glucose to help reduce blood glucose level in the fat condition. And insulin independent glucose uptake through group 2, right? Liver and pancreas beta cells. So actually in the fasting condition, the glucose transport activities should be lower, particularly glucose transport activity in muscle and adipose tissue, because of no insulin. Well, not no insulin, lower insulin. Yes, thank you. So because of a lower insulin, so group 4 activity is lower in muscle and adipose tissue. So we have a very low transport activities, right? So, yeah.
Is that the only one that's really sensitive to insulin?
You mean the group 4? Yes, yes.
The other ones aren't.
The other one's not.
Okay.
Yeah, it's not. So this also help to raise blood glucose level, right? You don't want to, uptake a lot of glucose in the muscle, so let more glucose remain in the circulation. But it's also is the mechanism, Okay, so let's look at how group 2 regulates glucose metabolism in fat condition. So in fat condition, group 2 basically mainly expressed in the liver and the pancreas beta cell, right? So in the fat condition, when we eat food and we raise the blood glucose level, and glucose can be uptaken by group 2 into the liver and also pancreas. So group 2 is the glucose dependent transporters. That means glucose level, high glucose level that can activate group 2 activity. So group 2 can uptake the high levels of glucose, right, into the liver and the pancreas. So what happens when glucose enters the beta cell in pancreas is to lead to insulin secretion, right? So beta cell basically is a glucose sensor. When glucose level is high after meal, then insulin secretion will be triggered, right? And what about in the liver? So liver can also uptake a lot of glucose in the fat condition by group 2 into the liver cells and to make glycogen, right, stored in the liver. And also glucose can be converted to fatty acids, and also use of fatty acid synthesis, lipogenesis, we're going to talk about later. So in the fat condition, glucose go to the liver for glycogen synthesis, store energy, and also converted to fatty acids, right? So this is how group 2 uptake a lot of glucose from the circulation into the liver and pancreas to release insulin and or to store glucose in the liver. So this way then can reduce the blood glucose level, right? So under the fat condition, so group 2, the role of group 2 is to transport the glucose into the liver for storage or into the pancreas beta cell for insulin secretion. So, any question? Yes.
What do you mean when you say glucose sensor?
Glucose sensor the sensor, when glucose level up, then this beta cell will be uptake a lot of glucose and active, to make insulin.
So that only be the pancreas, not the liver.
No, not the liver.
Okay.
Yeah. Well, I mean, in terms of insulin secretion, we call it sensor the beta cell. But if we're thinking about group 2, I think group 2 also, we can also call group 2 is a glucose sensor, right? Okay, so glucose stimulate insulin secretion. So if we thinking that our body responds to elevated glucose after meal, we can consider two different stage or two different sort of tier response. The first tier response or second tier response, right? So first tier response is elevated glucose can trigger insulin secretion, right? So this response we call first tier response to elevated glucose, right? So elevated glucose and stimulate insulin release. This is the first tier response in our body. So here I just wanted to tell you the process. This is the beta cell, right, in the pancreas. And again, the group 2 is the major transporter in the beta cells. And once glucose levels increase after meal, in the fat condition, glucose can be uptake into the beta cell and go to glycolysis pathway to make ATPs. And you have a lot of ATPs made, so increase the ratio ATP to ADP. And this increase will lead to the calcium channels open up and pump, and calcium can, potassium, right? Potassium can be, pumped out of the cells, and this will lead to depolarization of the cell membrane, right? And eventually, because of the depolarization membrane, so calcium channel is open, so calcium can be influxed into the cell and causing the increased concentration of calcium. So this calcium concentration increase is very important, can trigger the movement of insulin secretory granule and release insulin into the circulation, right? So try to remember this sequential process of how glucose stimulate insulin release, right? So again, glucose uptake by group 2 go to glycolytic pathway and make ATPs, increase the ratio of ATP to ADP, and then open the potassium channel and pump potassium out of the cells and causing the depolarization of the membrane, and this will lead to open the calcium channel and increase the calcium influx, right? So raise the calcium concentration inside of cells and then trigger insulin release, right? Any questions about this process? Okay, so the second tier response, right? So once we have insulin released from the pancreas in response to the increased blood glucose level after meal, right? So and insulin then in turn do something to further reduce blood glucose level, right? So insulin is the hormone that regulates transport activity, particularly transport 4 activity in muscle and adipose tissue to increase transport of glucose from blood into the muscle and adipose cells, right? So this process is called second tier response. So secondary response basically is insulin stimulate glucose transporter into muscle adipose tissue by group 4. So I hope this will be clear. This two-tier response, right? First tier response is basically the dietary glucose entered, absorbing to the circulation, then can trigger insulin secretion, right? This is the first tier response. And insulin stimulate glucose uptake into muscle and adipose tissue by group 4 is a second tier response, right? Now, glucose transport and tissue are involved in the second tier response, glucose metabolism. So here's the 3 blanks, like you to fill in. So which glucose transporters that involve the second tier response? Group 4, yes. And which tissue that involved in the second tier response? Yes, muscle and adipose tissue, right? So what about the first tier response? Which group 4? I don't have a slide here, but I'm just asking you. Which glucose transporters are involved in the first tier response? Group 2. Which tissue that involved first tier response? Beta cell, right? Pancreas? And, pancreas. Yes. Okay. All right, so I guess we have an informal writing, right, assignment today. I do have actually, let's see. I have a question. I do have a question, but I don't think I have covered this material, so I want to make a change. Let's do this for the informal writing for today, right? So we already explained, right, group 2 and group 4 was very important transporter, right, in terms of the regulation of glucose homeostasis. So the question for this informal writing is what happens to glucose homeostasis if group 2 or group 4 is defective, right? Yeah, you guys work together. I mean, open book discussion, you can work together, but you need to turn in individually, right? You work together, but you...
So this is the first year.
Oh, one thing I just wanted to mention that in terms of classroom, Sarah helped her to try to work out and whether we can move to the 15, but she's still working on it. So, I mean, we probably just stay in this classroom until I have a solution. Sorry, I'll just do a quick thing.